Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 6 for October 2022. Let us begin with the first question. Question 1. The photograph shows an aquatic plant, Kabomba caroliniana. So this is the aquatic plant. They say these plants live in ponds, lakes and slow-flowing rivers in North and South America. If the stem of the plant is cut underwater, bubbles of oxygen gas are released from the cut end of the stem during photosynthesis. Moving on. Here they say, describe an experiment to investigate the effect of temperature on the rate of photosynthesis in Kabomba caroliniana. So here the independent variable is temperature. We will set up experiments in beakers at least 5 different temperatures. For example, it could be 10 degrees, 20 degrees, 30 degrees, 40, as well as 50. And then we'll cut the plant stem underwater and connect to a photosynthesometer with a cut end towards the funnel connected to the photosynthesometer. Then we have to use excess sodium hydrogen carbonate. Remember, this provides CO2, so this ensures that carbon dioxide concentration does not limit the rate of photosynthesis. Then we have to use aluminium foil on the other side of the beaker so that the plant is exposed to light from one direction. Then place the light source at the same distance in all experiments. This ensures that the light intensity is the same. Then collect the gas produced in a specific period of time, for example 10 minutes, and then calculate the rate by dividing the volume of oxygen produced by the time. You remember to allow the plant to acclimatize. This is if you're using the same plant for all the experiments, you have to allow the plant to acclimatize before results of the next temperature are collected. Then repeat the experiment at a temperature and calculate the mean. Here we have to control the pH using a buffer, and we have to control the wavelength of light using the same color filter. Moving on. In part B, describe how oxygen is produced during photosynthesis. Oxygen is produced through the process of photolysis. For example, here, in photolysis, water is split into 2 hydrogen ions, electrons are produced, and an oxygen atom. But in the process, 2 oxygen atoms are going to come together to produce an oxygen molecule. So I said, photolysis leads to the splitting of the water molecule, release of hydrogen ions, electrons, and an oxygen atom. Then 2 oxygen atoms combine to form an oxygen molecule. Examples of reactions are that or that, but they were not required. We just had to describe in words. So this brings us to the end of question 1. Let's continue to question 2. Question 2, the photograph shows a cane toad, Rhinella marina, which is the toad here. This amphibian is an invasive species in northern Australia. Pesticides can kill these amphibians quickly. The outer layer of amphibian skin is only 1 or 2 cells thick. This compares with the skin of mammals, which is 20 to 30 cells thick. Caffeine is a chemical with similar properties to pesticides. A scientist investigated the diffusion of caffeine across skin samples from a cane toad. Caffeine solution with a concentration of 200 milligrams per decimeter cubed was added to the top chamber of a diffusion cell. Samples of solution from the bottom chamber were removed every hour and the concentration of caffeine measured. The diagram shows a diffusion cell used to measure the rate of diffusion across a skin sample. So in this diffusion cell, we have this upper chamber where the 20 centimeters cubed of caffeine was added, and then we have the skin sample here. Then there is a sampling tube to remove some sample in order to measure the concentration of caffeine. This is where water comes in, and that is where water leaves. Moving on. Here they say the graph shows the results of this investigation. So on the vertical axis, we have concentration of caffeine in the sample, which is in microgram per centimeters cubed. And on the horizontal axis, we have time in hours. We can see from this graph that as time increases, the caffeine concentration in the sample also increases. However, we see this is the greatest increase in the concentration of caffeine in the solution that is being sampled. So down here they say, describe how 20 centimeters cubed of caffeine solution with a concentration of 200 milligrams per decimeter cubed could be prepared. Here I try to calculate to show you how I obtain my 4 milligrams. We know we are making 20 centimeters cubed, which is the same as 20 times 10 power negative 3 decimeters cubed. Remember, this is milligram per decimeters cubed. So using mass concentration is equal to mass divided by volume. So the 200 is going to be mass divided by the volume, which is this one here, 20 times 10 power negative 3. So making mass the subject is going to be that times that, which is what I showed here, and my answer was 4 milligrams. So we're going to dissolve 4 milligrams of the caffeine sample into 20 centimeters cubed. So my answer was add 4 milligrams into 20 centimeters cubed of the ionized water to create the solution. Here they say calculate the rate of diffusion per minute between 1 and 2 hours. So between 1 and 2 hours, we had to find the gradient of that line. You can see I got the coordinates for this point, which is 124, and here we have 280. So the rate is going to be 80 minus 24 divided by 2 minus 1, which is going to be 56 microgram per centimeters cubed per hour. However, they want us to find the diffusion per minute, so I divided that by 60 because 1 hour is equal to 60 minutes. So 56 divided by 60 gave me that, which I rounded off to 0.93 as my answer. Here they say the rate of diffusion between 4 and 5 hours is 0.4 microgram per centimeters cubed per minute. Calculate the percentage decrease in the rate of diffusion between 1 and 2 hours and between 4 and 5 hours. Between 1 and 2 hours, the rate was 0.93, and here we have 0.4. So 0.93 minus that gives us 0.53. So the percentage decrease should be 0.53 divided by the original times 100, giving us 56.989, and I rounded off to 56.99%. Next they say, caffeine is a polar molecule. Explain the difference in the rate of diffusion between 1 and 2 hours compared with the rate of diffusion between 4 and 5 hours. So the fact here we can see between 1 and 2 hours the rate was higher than between 4 to 5 hours. So the rate of diffusion is higher between 1 and 2 hours because caffeine, a polar molecule, is transported through the channel proteins by facilitated diffusion down a concentration gradient. However, over time, the concentration gradient decreases, so the rate of diffusion also decreases. Moving on. In part B, state one abiotic and one biotic variable that could affect this investigation. For temperature, this is because molecules that are diffusing require a high kinetic energy for diffusion to be faster. And for pH, this is because some channel proteins could be affected or they could be denatured, and the rate of diffusion could decrease. Then the biotic variables include sex as well as gender of the toad. Here they say choose one of the variables you have identified. Describe how this variable could be controlled and the effect it could have on the results if it's not controlled. The variable I chose was temperature, and we can control this using an incubator, or you could use an air conditioner or a thermostatically controlled water bath. And if this is not controlled, the results will not be valid. So this brings us to the end of question 2. Let's continue to question 3. Question 3. The photograph shows one variety of wheat and the grains it produces. So this is the wheat and the grains. They say a scientist observed that the germination of wheat grains was affected by the presence of arsenic ions in the soil. Groups of 30 grains were placed on a filter paper in petri dishes. Each petri dish had 0.5 centimeters cubed of water with a different concentration of arsenic ions added. The petri dishes were incubated at 23 degrees Celsius for 4 days. The percentage of seeds that germinated were recorded. The procedure was repeated a number of times. The table shows the results of the investigation. Here we can see as the arsenic concentration increases, the mean percentage of seeds germinated after 4 days decreases. From the table, standard deviation was included, and this shows that the experiments were repeated. So down here they say, identify one risk associated with this investigation and how you could reduce this risk. Because we are working with plant materials, there is a risk of allergic reactions. And how can we reduce this? By wearing gloves and a lab coat. The next they say, state a suitable null hypothesis for this investigation. There is no correlation between the mean percentage germination and the concentration of arsenic ions. Next they say, plot a suitable graph to show the effect of the concentration of arsenic ions on the percentage germination of wheat grains. So here, if I make it smaller so that you can be able to see clearly, on the vertical axis, I place the mean percentage seed germination after 4 days and the units are percentages. And then on the horizontal axis, we have the concentration of arsenic ions. The units are milligram per decimeters cubed. Here I placed error bars for each of the results. You can see the data sets. And then I connected the point using a suitable line. Moving on. In part B, the scientists used a statistical test to analyze the data. To calculate the correlation coefficient, the scientists produced this table. So we can see they gave us all the information, including the d's, but we were not given d squared. So down here they said calculate the correlation coefficient RS using the formula, which is that, where this means sum of, d is the difference between each pair of ranks, and n is the size of the sample or number of pairs or values. So if this is d squared is 0, d squared is 1, d squared is 1, d squared is 0, d squared is 0, d squared is 1, and that is 1. So when we add up everything here, we get the summation of d squared is equal to 4. So this part is going to be 6 times 4. And then n, we have to count 1, 2, 3, 4, 5, 6, 7. These were 7 samples or 7 data sets. So I used n is equal to 7. Now in my calculation, RS is 1 minus 6 times 4 divided by 7 into 7 squared minus 1. And using a calculator, it was 0.92857,143. Rounding it off, it became 0.93, which is the answer here. Next, they say the table shows some critical values for this statistical test. So they say comment on the conclusions that can be drawn from this investigation. Use your graph, your calculated RS value, and the table of critical values to support your answer. So remember, the critical values we use in biology is the one at p is equal to 0.05. So since there were 7 number of pairs, the corresponding value is going to be this one as our critical value. So we can see comparing 0.786 and 0.93, which was a calculated value, this is lower than the calculated value. So here I said the critical value is 0.786. Since the calculated value, which is 0.93, is greater than the critical value, we reject the null hypothesis. So there is a correlation between the mean percentage germination of seeds and the concentration of arsenic. However, error bars overlap, so the data is not valid. If you can go back here, we can see there is great overlap in the data sets, so we can say the results are not going to be valid. This overlaps with that, and this overlaps with that, so we cannot know if the germinations at different concentrations are due to the variations or there is an error that was carried out during the experimentation. In part C, in breeding experiments, scientists select varieties of wheat with different characteristics. Explain why some varieties of wheat have different tolerance to arsenic ions. Because they've told us these are different varieties, it means they have different alleles, and the different alleles could enable the plant varieties to resist absorption of arsenic. So I said different varieties have different alleles. The alleles could enable different response towards arsenic. Some varieties are unable to absorb arsenic during germination, meaning they will not be affected. The next part says, explain how this investigation could be extended to study the effect of arsenic ions on growth of wheat plants from germinating wheat grains. The aim here is to study the effect of growth, not the effect of germination. So what we need to do is germinate some seeds without arsenic, and then after germination, expose the young plants to arsenic of different concentrations and see how that is going to affect the growth. So I said germinate seeds in absence of arsenic, then grow the germinated seeds in solutions of different arsenic concentrations. In the experiments, control temperature, mineral ions, sunlight, pH, and any other growth factor. Then measure the height or mass of the produced plants before and after exposure to arsenic, and calculate the change in height or mass after a period of 1 month. Acceptable time is between 7 to 100 days. So this brings us to the end of question 3. Let's continue to question 4. Question 4. The photograph shows a polychaete, which is that one in the Indian Ocean. Down here they say, this animal can be kept in tanks in a laboratory. This animal uses a fan to feed on organic particles suspended in seawater. When the fan is touched, the fan is withdrawn into the tube by a reflex action. The fan then extends to allow the animal to continue feeding. A student observed that some of the animals withdrew their fans completely, while other animals showed very little reaction to touch. This means they are somehow habituated. The student formed the following hypothesis: the greater the number of touches, the less the animal reacts. So here they say plan an investigation to find evidence to support or reject this hypothesis. Describe preliminary practical work that you may undertake to ensure your proposed method will provide quantitative results. Because this is starting habituation and the organism removes the fan in order to eat or to feed, we need to find the suitable time when this organism can be able to feed or this animal can feed. We need to find suitable food that this animal is going to eat. We need to find suitable ways of touching the animal in order to stimulate it into action. So I said, find the suitable conditions for the animal to feed, find a suitable time for the animal to feed, then find a suitable method to touch the animal, and find a suitable method to determine if the fan is withdrawn. So, and to what extent is the fan withdrawn? Moving on. Here they say, devise a detailed method including how you would control and monitor important variables. So here we have to find how we're going to vary the dependent variable, how we're going to measure the dependent variable, how we're going to keep some conditions constant, and in what time is the experiment going to be carried out. So I say the dependent variable is the extent of fan withdrawal. Here we need to supply organic material to the water tank to enable the animal to feed. We need to touch the animal with a cotton bud. This is so that it is not hurt in the process of touching it. And then in doing so, ensure that the touches are with the same force or same strength. Then allow the animal to acclimatize before the next touch. Touch the organism a specific number of times in a specific period of time. This is to see if repeated touches cause the withdrawal or not. That tells us if the animal is habituated or not. So in this case, this is like measuring the dependent variable. Then control the temperature using a water bath. Also ensure that organisms used are the same gender. This is if you're repeating the experiment with another organism. Then control light intensity, water vibrations, and ion concentration of the water. Then repeat the experiment with another animal and calculate the mean. In part C, describe how your results should be recorded, presented, and analyzed in order to draw conclusions from your investigation. So here I did not draw any table or graphs, but I just wrote. And again, to clarify with you guys, you are not required to draw a table or a graph here. You can just write the information down here. You'll be okay. So I said record the raw data in a table with headings and units, and then include a column for mean calculated from repeat experiments. Then draw a line graph or scatter graph with labeled axes and units. Then use a suitable statistical test like Spearman's to conclude if a correlation exists. Lastly, suggest 3 limitations of your proposed method. The limitations here are it's going to be hard for us to know if the fan is completely withdrawn, it's going to be hard for us to touch the animal with the same strength every time, and it's going to be hard for us to know if the organic matter we have put in the water is equally distributed. So I say it is difficult to touch the animal with the same strength or force. It is difficult to determine the extent of withdrawal of the fan. It is difficult to know if the organism is already habituated or not. This is before you begin the experiment. We cannot know. Even during the experiment, it could be difficult. So it is difficult to ensure that water currents or vibrations and distribution of organic material are the same throughout the water tank. So this brings us to the end of question 4 as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.